Today, I have the privilege of introducing our next speaker. So this is Garam Kim. She is a postdoctoral fellow in Evan McCoskow's lab at the Broad Institute and a Leffler Fellow at the Leffler Center for the Study of Neurodegenerative Diseases at Harvard. She previously completed her PhD in neurosciences in Aaron Gilter's lab at Stanford University. Her research focuses on developing technologies that combine high-resolution imaging and high-throughput genomics tools in a spatially resolved manner. She hopes to utilize these spatial tools to interrogate the biological underpinnings of neurodegenerative diseases and to enable people from diverse fields to study their own research questions using these tools as well. Today, she's going to give us a primer on spatial transcriptomics technologies. Thank you. Oh, she'll take questions anytime, and thank you.
Can everyone hear me okay? Okay, perfect. Thank you all for being here at this early and rainy hour. And thank you both to the organizers for inviting me to share this primer on spatial transcriptomics technologies. And as she just said, please feel free to stop me at any point. I will do my best to answer any questions that you may have. Okay, so to give you an overview of this primer, I'll first give you a brief introduction on spatial transcriptomics, what it is, key concepts, and why we want to use these technologies. Then I'm going to walk you through some examples and how we can broadly classify these technologies. And then we'll discuss each method's strengths and challenges associated with these types of technologies, as well as some practical considerations for what to look for when you're choosing which tool you may want to use for your particular study. And lastly, we'll touch on some immediate and medium-term future directions in the field of spatial transcriptomics. Okay, so what is spatial transcriptomics? I think that to understand fully spatial transcriptomics, we first want to understand its most famous predecessors, in particular, bulk RNA sequencing and single-cell RNA sequencing. So this is a cross-section of a human brain. And as you can see, there are many Lego pieces that define different distinct brain regions, and perhaps even distinct structures at the micro level. And what bulk RNA sequencing looks like is like this. So when we sample any piece of tissue from this human brain, basically we grind up all the RNAs in a way that's kind of indistinct and disorganized, because now it's kind of in a soup of Lego blocks. And this is advantageous in a way because it's very high sensitivity. You are just grinding up the RNAs and sequencing all the transcripts that are there. However, you're losing a lot of information because you're averaging all the gene expression across a sample. So you can't tell if an RNA transcript came from this Lego block versus a different Lego block. Okay, then the next, I guess, big technological advancement was the development of single-cell RNA sequencing. And so what this enabled was that now we could look at individual cell types and cell states. So now you can see that the Lego blocks are kind of separated out in space. It can be grouped and you can kind of see what structures they came from. But one thing that you still lose is actually its context and where it was in space. And this is because in order to get these single cell or single nuclear suspensions, you have to dissociate the tissue. So then finally, this brings us to the advent of spatial transcriptomics, which preserves tissue structures, and it allows you to kind of see, okay, what did all of my neighbors look like, and what are the kind of cell interactions and microenvironment that surrounds that particular cell. Okay, so why do we care about studying gene expression in space? Well, first, this is very small. But this is an example of a very earlier UMAP plot from one of the earlier spatial technologies. And what this is it's a UMAP plot is a type of plot that you get in single-cell RNA sequencing, where every dot represents unique cell or nucleus. This dimensionality reduction allows us to see that cells that are closer to each other in their transcriptomic profile will tend to be also closer together in space. And so based on this unbiased hierarchical clustering, we see that there are distinct cell types that are perhaps associated with distinct regions. Now, the moment you put this into space, what you can see and begin to appreciate is that across these different tissue types, you now start to see, rather than just random dots in space that kind of cluster together, now you can now start to see where they lie in space and what its neighbors look like. And you can even see that these ependymal cell layer, for instance, it's a single cell layer right near the hippocampus in the mouse hippocampus. So basically, studying gene expression in space allows us to see what cells is it interacting with? What does the tissue architecture look like? If there's a disease microenvironment, is there any kind of inflammatory niche? Is there some kind of cell population that's going to, I guess, aggregate together? And we can start to see the spatial patterning that tells us a lot more about what's happening biologically. Okay, so in addition to the sequencing-based technologies, I thought it would be nice to look at how we came to the state of the spatial genomics field as we know it today. And there are many historic milestones and so many technologies that have been developed over decades. But the way that I have tried to kind of condense it is, I guess, to the particular experiments or developments that really pave the way for modern the modern technologies that we know today. So starting in 1969 is actually when we saw the first ever in situ hybridization. So this was the first technique in situ, so in place, in the tissue, without having to isolate anything, that they were able to use radioactive probes that would label a very specific DNA sequence. And so you can see it labeling in little dots inside the nucleus. And this established the principle that you can actually detect nucleic acids in a native context. Another year and I guess technology that I chose was 1996, the invention of laser capture microdissection. And the idea was that you could use a laser to basically cut very focal fields of view inside a tissue and transfer it onto this film. And then you would be able to do whatever downstream analysis you wanted to do. So at this point, what this shows is that you're able to kind of preserve some level of spatial information. And this is a big step towards spatial precision. And in this case, this isn't a specific cell. This is actually beta amyloid plaques that's associated with Alzheimer's disease. Then okay, so actually I flipped the order of those, so please forgive me. So the other, I guess, major development was not just in situ hybridization using radioactive probes, but now using fluorescence in situ hybridization, which introduced this idea that now you can have multiplex spatial transcriptomics. So you can now visualize more than one type of transcript at a given time. And finally, this brings us to modern day spatial transcriptomics that we know today. So higher throughput technologies that now allow us to visualize many, many transcripts inside of cells using very clever strategies. Or I guess in this context, this is a single cell with many transcripts labeled inside. In the case of the right image, this is one of those UMAP plots that I described earlier, but now plotted out in space, and we can see that from a single experiment that all of these different cell types are able to resolve. Okay, so what are the types of spatial technologies if we're going to broadly classify it? So this is not going to be a comprehensive list because there are way too many technologies, but I'm going to highlight a few, especially a few that are commercially available or perhaps were invented at the Broad. Okay, so again, there could be many ways you could classify it. I think one way that I decided to classify it is into two broad categories of spatial technologies. So one of them is imaging-based spatial technologies, and the other is sequencing-based. So in imaging-based methods, the RNA is always detected through repeated cycles of fluorescence imaging. So you would have some kind of targeted probes that target a sequence that you're interested in. You would fluorescently label it. And there's a limited number of channels that you can image from at one time on a microscope. So people have figured out pretty clever ways to really scale this up so that you can target, around 10,000 species at a time, even though most people choose not to, but that is definitely possible. And one really nice thing about having this image-based readout is that you have really high resolution. You see every molecule, so it's very, very, like you can see subcellular structures, you can see the cell morphology, and things like that. Sequencing-based methods are different in that you never actually look at the image of your tissue. I mean, you could acquire that, but the actual readout of the RNA itself is that the RNA is captured basically off the tissue or through tissue dissociation and then sequenced. And in this case, you don't require designing specific probes to target transcripts of interest. So you have a non-targeted capture method, and this actually enables whole transcriptome-wide profiling. In terms of the resolution, that is dependent on technology, and we'll get into the different types of technologies that are available. Okay, so thank you. Okay, so in terms of subclasses, these are two very broad categories, but I thought I would divide them further into a couple subclasses. So for imaging-based technologies, there's a fluorescence in situ hybridization that I mentioned earlier, as well as in situ sequencing. And I'm going to go into what those both mean in greater detail. And in terms of sequencing-based, perhaps we could divide it into things that technologies that use a spot-based, so some kind of barcoded array that already has defined spatial positions with spatial barcodes. And then that captures the RNA from the tissue, or cell-based, where you are actually tagging the cells with the spatial information and then using that to put it back into space. Okay, so let's now go into examples of imaging-based spatial technologies. These are just a few that I'm going to mention. But first, I guess for any imaging-based spatial technology, if you look at the key principles, there's basically three major steps. So the first step is that you're generating some kind of optical barcode. So some kind of barcode that you can use to identify the particular RNA transcript of interest. And that barcode is different across the technologies, and I'm about to get into that. But generally, you'll have some kind of probe that's binding to a target of interest. And then I guess you're generating fluorescence signal in different ways across different technologies. And then you read out this signal via single or multicolor fluorescence. And then once you have that fluorescence signal, then you would image and then get rid of that signal and then do another round of imaging. So you have this constant cycle of sequential imaging followed by signal removal. And you do this as many times as necessary for your particular set of transcripts that you're probing for. And finally, once you have acquired those series of images, then you're going to have to use the actual images themselves to figure out which fluorescent spots correlated with which transcripts over the rounds of imaging. And so this is how you're going to then identify what was the actual identity of that RNA transcript. Okay, so two of the, I guess, two very commonly used fluorescence in situ hybridization methods, FISH methods, are called MERFISH and SEQ-FISH. So MERFISH stands for multiplexed error-resistant fish, rolls right off the tongue. And SEQ-FISH is sequential fish. So I think that kind of is self-explanatory. So I mentioned that for each of these technologies that they have a different way of generating the optical barcode. In the case of MERFISH, it's a binary code. So which means every transcript is actually just given a random and unique series of zeros and ones. So So you will never have two overlapping series of zeros and ones. So let's assume like transcript 1 has like the sequence 1100, and RNA 2, a different transcript, has the sequence 1001. That would mean that for the first round of imaging, you would have probes that target these two transcripts. And in the next round of imaging, you would be missing a fluorescence readout for the molecule that's binding that particular transcript. Sequential FISH is actually a very similar idea, except that instead of a binary code, now you have a color code. So how does this look? So this is a cell, if you couldn't tell. And here are 5 different transcripts that are being imaged in this round. And so this is just at the first base position. Now we're going to image and figure out which fluorescent spots had a 1, so there was signal. And in the next round, perhaps this transcript now had a 0 in that spot. And so you're imaging the same spot over all of these rounds of imaging until finally, once you acquire all of the images, you figure out what was this string of 1s and 0s at that particular place. And then you can decode it to basically figure out what was the identity of that transcript. And then again, sequential FISH is kind of similar in idea, except that this time they were given different colors instead of a binary 1s and 0s. Okay, and then what about the other kind of subclass, broader subclass under imaging-based spatial transcriptomics methods is it's called in situ sequencing. So 2 of these methods are, one's called in situ sequencing, and the other one, and you might know that one better as Xenium from 10x. And the other one, this was developed by a faculty member also at the Broad, Shaw Wang, is called StarMap. Another one that rolls right off the tongue called spatially resolved transcript amplicon readout mapping. So I mentioned that MERFISH had a binary code and sequential FISH had a color code. In the case of these in situ sequencing based methods, they have just a unique nucleotide barcode. So a series of ACs, Ts, and Gs. And what this class of technologies relies on is a technology called, I guess, or type of circularizing of DNA called padlock probe hybridization. So you have a padlock probe is basically a single strand of DNA, and you have these sequences at the ends that are complementary and completely next to each other on that's targeting a particular strand of interest. And then once it binds, then it'll circularize. Then you have a ligation step to basically get rid of that gap. And now it's fully circularized instead of just bound like that. And then there's the other main concept here is called rolling circle amplification. And what that means is that DNA polymerase will bind at a priming site. And then it'll just basically circle around and around and around until it's just like super amplifying itself. So in situ sequencing and StarMap both use this padlock probe plus rolling circle amplification technology. And so I guess, yeah, so there's going to be some reverse transcription at some point, and the way that is done differs. There are different nuanced, I guess, approaches between the two technologies, but the basic idea of padlock probes and RCA is there. And then finally, how do you actually read out the unique sequences that those probes bound to? So there's going to be some sequencing by ligation. So basically, if these barcodes are 4 to 7 nucleotides long, then there's going to be some kind of sequencing primer, then a ligation of a fluorescence-based nucleotide that's going to read out what was that nucleotide at that position. So it is truly like you're actually sequencing out the actual sequence, unlike MERFISH and seqFISH. Okay, finally, I guess the last in situ sequencing method that I thought I would talk about is called expansion sequencing. And for those of you who are not familiar, expansion microscopy is a very cool technology. So, not everything, is resolvable in space with high definition. So if you have something very small and you have a microscope that doesn't reach that resolution, how do you fix that problem without just making a fancier microscope? Is that actually, instead of making the microscope zoom in more, you can actually just make the sample expand. So it's artificially bigger. And the way that they do this is by embedding the tissue inside of a hydrogel and then adding water, and it'll literally expand. So what you're seeing here is that there are some cells, and now they've embedded it inside this gel, and now it's expanded. And then now you can do in situ sequencing as you would have done otherwise. The other thing that was in this expansion sequencing paper is that you could also do ex situ sequencing. So basically, after you've sequenced it in situ, if you wanted to have an additional quality control, you could basically isolate out the RNAs that you created in situ and then sequence it out, externally. Okay, yeah, so basically the 3 key steps are expansion, in situ sequencing, and then optionally ex situ sequencing. That brings us to sequencing-based spatial technologies. So there are also a lot of them. I won't talk about every single one, but that's just some listed. And again, the key principles that are shared across all capture and sequencing-based technologies. In terms of capture-based technologies, the idea is that you have some kind of capture array that has some kind of spots that could be like literal spots, it could be beads, it could be some kind of cluster. But each spot has a unique set of spatially barcoded DNAs. And these barcodes have a unique spatial barcode per one of those spots. It has a unique molecular identifier. So if you have multiple molecules that share the same spatial barcode, that can be captured. And then it has a poly-DT at the end. So that'll allow us to capture all polyadenylated mRNAs in a very unbiased manner or untargeted manner. And then, yeah, so, and then I guess, so the key principles, in addition to the use of a capture array, is that you're going to place a piece of tissue. It could be fixed or frozen, and then you place it directly on top of the barcoded capture array. Then you would actually capture the RNA. So perhaps the RNA will diffuse onto the array and bind to the spatial barcodes, or the other way around. Then you're going to remove the tissue and prepare, I guess, you'll have some kind of reverse transcription to get cDNA, then you'll prepare the libraries for sequencing, and then you'll actually sequence it out. So that's the basic principles. So now going into examples of major capture and sequencing-based methods is, I guess I put these 3 on the same slide because I guess you can kind of see side by side how they're quite similar, but the ways in which they differ. So 10x Genomics has a product called Visium. So basically the original version has a glass slide that has this kind of barcodes at a resolution where the beads are about 100 microns apart, center to center. And so you can imagine that's actually on the lower side of resolution now in retrospect, because it's not single cell resolution. Single cells are much smaller than that. However, since then, there's been Visium 2, which has been adaptable for fixed tissues, and Visium HD is the latest version that now gets actually down to 2 micron resolution, and they use squares that don't have any spacing in between, so it's supposed to be a lot higher resolution. And it's compatible with formalin fixed, paraffin embedded, or FFPE tissues. So actually, tissue fixation is another topic that we'll come back to in a little bit. Then there's this high definition spatial transcriptomics, so called high definition ST. So ST, which was the version before this, is actually, I think, the first spatial high throughput spatial technology. But the idea here is that you have these wells, and each of these wells has a single bead, and each of the beads has a unique spatial barcode. And so this bead is about 2 microns in size. So that's where the high definition came from. And then SlideSeq is a technology that was developed in my lab, which is Evan McCosko's lab, in collaboration with Fei Chen's lab, also at the Broad. And the way that these capture arrays are created is that we have a bunch of 10 micron beads that are that we can basically spin down onto glass slides or a glass surface, and it'll just be randomly situated, and we will sequence those so that we have a ground truth of where the spatial barcodes are at each of those locations. Then we place a tissue on there, allow the RNAs from the tissue to diffuse and bind onto these bead capture the poly-DTs here, and then we'll do the downstream processing to get the libraries prepared and sequenced. Okay, and then another technology that also rolls off the tongue is called dbit-seq. So this is a little bit different from the capture-based arrays that we just talked about. So dbit-seq stands for deterministic barcoding in tissue for spatial omics sequencing. And instead of bead or wells, this actually, or having this kind of array, this is actually a microfluidics-based method. So basically you have microfluidics chips that have parallel lanes throughout, and you clamp this microfluidic chip directly onto the tissue. And then into the microfluidics wells, you're gonna introduce poly-DT tagged DNA barcodes inside those channels and let it just flow over the tissue. Then you're going to add a second, I guess, perpendicular microfluidic chip on top. And then what you put into that is a biotinylated second barcode that's going to have your unique molecular identifier information, and it's biotinylated so that downstream it can bind to streptavidin beads and be used during library preparation and generation.
Can I ask you a quick question
Yes.
about that? Are there plans to make this 3-dimensional so that you get the XYZ position of these barcodes?
So I think that is a common goal across all the technologies. StarMap is the only one among the technologies I've discussed where in their initial study, they kind of presented it as a 3D method. One thing I didn't mention is that StarMap has hydrogel embedding as well, and they do very mild expansion and also tissue clearing to kind of help with the imaging. In the case of dbit-seq, I don't believe that is it's there yet, but I think all technology at the end of the day are striving to get there at some point. Yeah, thank you for that question. Okay. So now getting to higher resolution, this is called StereoSeq, so spatial enhanced resolution omics sequencing. And what this type of array uses is called DNA nanoballs, which is DNB. So basically what you do is you have single-stranded DNAs, and you also use rolling circle amplification to just make like a bunch of copies of itself. And then because DNA is very negatively charged, it'll just kind of like spontaneously like go into like this ball, which is why they have all of these balls. And then they have a pattern chip, and basically they'll flow over this DNA nanoballs across all of the flow cell. And because these nanoballs are about 200 nanometers in size, it'll naturally kind of only occupy one per pattern spot. And then that's actually used to capture the RNA. And then again, similar principle downstream, but that's primarily the difference with other technologies. And I believe this is the last capture-based method that I'm going to mention. It's called OpenST. It's open because like everything, their scripts and their methods and everything is very open. They want everyone to use it as open source technology. But they had a very clever idea of taking the Illumina flow cells, which have very, very high resolution, and it's very tightly packed. And they took advantage of their sequencing by synthesis method to basically, within the flow cell, generate unique spatial barcodes there. And then they would do some kind of post-processing to make it amenable to RNA capture. And you can basically break up, they have like you can cut these flow cells. They have a cutting like, like some technology that they say you can also do. And then basically in terms of the size that you're able to spatially profile is going to be limited to the size of these flow cells. So you can see actually in length, it's actually quite good. I think I forget exactly how many centimeters, maybe like 9 or 10 centimeters or something like that. But in width, it's, small. So, but I think this is a really exciting idea. And again, you're going to have the RNAs bind to that flow cell. And yeah, and you're able to then get very high resolution information. So that was all spot-based. But the second subclass I mentioned, and I'm actually only going to give one example, and this is the technology that is most dear to my heart because my postdoc project is built off of this technology. So if you have any specific questions, let me know. So what I mean by cell-based is that instead of capturing RNAs onto an array, we're actually now tagging the cells with the spatial information. And this method is called slide tags. And so slide tags actually still uses a bead array as well. And they also have spatial barcodes. But the difference is that now it has a photocleavable linker so that when you shine UV light on it, the spatial barcodes will cleave off of the bead array and tag the tissue. And so you just have an intact piece of tissue. And once the nuclei are tagged, then you can actually isolate the nuclei. And then you can use any established single cell profiling technology beyond even just the transcriptome to profile these nuclei in a very high throughput fashion. So it basically, I guess, what's the word? Tail? Anyway, it'll blend in to existing workflows very easily because all you're doing is just adding a tag of DNA barcodes and then you just remove the array. So then I guess the advantage of this is that now you have the very high throughput capture, untargeted capture of RNAs. So you can still get this high resolution UMAP. And then at this point, using the spatial barcodes that each nucleus is tagged with, you can now map it back into space, depending on what were the spatial barcodes and in what quantities it was tagged with. So now when we're actually comparing all of these spatial methods, you can see that the resolution, the spatial resolution across these technologies differs. This figure is taken from a review that was published in 2022. So it's definitely missing some of the very big technologies. But you can see that even at that time, we were seeing 0.25 micron resolutions. Yeah, but one thing I wanted to mention is that so far we have talked about spatial resolution and how small are these spots that we can actually tag the cells with. But actually, at the end of the day, that's a spatial resolution. But what we haven't really discussed is the RNA resolution. And at least on the RNA end, at the end of the day, single cell sequencing remains kind of the gold standard for unbiased transcriptomics. It's very reproducible. It's commercially available. There's a massive computational toolbox. It covers genome-wide transcripts. And there's also established multimodality in terms of what other information you can capture, especially I think one of the most common ones is the RNA and the TAC-seq multiome. And one way to really drive this home is a direct comparison of the RNA profiles and UMAPs across 4 different technologies, 3 of them capture-based and 1 of them cell-based. And so you can see that, again, I mentioned at the beginning that when you have this dimensionality reduction, cells that are closer to each other in their gene expression profile will also be closer to each other in UMAP space. And the more clearly that they isolate from each other, I guess, the more distinct they are. And when you also look at, various cell types and gene expression, you can see, for instance, in the Xenium data set, that when you look at an excitatory neuron marker across all the clusters, that perhaps it's looking like it's expressed in a lot of clusters. And part of that is that actually the spot, no matter how big or small, you can have nanoscale resolution in a spot, but you still have to actually map it back to the cells. So it's actually a very tricky computational problem to try to deconvolute what the signal from each of those spots and which cell it came from. So that's just another thing to keep in mind as we consider these technologies. So now we'll briefly touch on the strengths and challenges of each of these methods and hopefully have a practical how to really choose the platform that's for you. So the strengths and challenges of imaging-based methods, as you saw, you can really directly visualize every single fluorescence molecule. And so that subcellular resolution is really nice. I think there's something very beautiful about being able to see the morphology and the cell structure and everything around it. So you get the morphological context. You can also co-detect it with protein. And you don't have any kind of tissue dissociation artifacts because you're looking at the tissue as is. However, in terms of potential challenges is that requires very specialized microscopy equipment. You will be limited to targeted gene panels because you have to always design the probe. You do have a kind of molecular crowding situation where if you have too many molecules together in space and you have fluorophores that are bleeding into each other, that might pose a small challenge. With expansion, I think it's a little bit more resolvable, but nonetheless, that can be a challenge. Imaging-based methods also will pick up all fluorescence signals, and that's not limited to specific fluorescence signal. It will also pick up background fluorescence. And particularly in human tissues, there's gross amounts of autofluorescence that come from lipofuscin buildup over time. So that is also another challenge to keep in mind. Given that there's this probe hybridization, stripping, and rounds of imaging, these protocols can be very complex and require very long imaging times. And that can also sometimes lead to photobleaching and light toxicity. So that's also dependent on how many rounds of imaging, what does your panel look like, how many transcripts are you trying to probe, but that is a potential challenge. And with each of these probe designs, you need it to actually be all at the same time. You need to be very highly specific. You don't want it to cross-react because they are kind of short oligo sequences that are binding to your transcripts of interest. So you want to make sure that it's not going to have off-target effects and bind to other similar transcripts. So that's what we mean by minimal cross-reactivity. And then you want to make sure that the hybridization efficacy is pretty uniform across all the transcripts. You don't want one probe that's going to bind much stronger than another transcript. And finally, to follow every single one of those dots and figure out, keep track of their identity, it can be very computationally intensive.
I may have missed this Are all the methods poly-T RNAs, or can you capture non-poly-T RNAs?
You can, so, so one of the technologies that I use is actually, well, so 10x has this flex kit that has probe-based capture, but it captures a whole transcriptome. And depending, you can just basically change out the handle so that it's binding to whatever transcript of interest. I think poly-A RNAs are easy because they're all, regardless of transcript identity, they all have A's. But I think there are ways to enrich or deplete for specific types of RNAs or sequences that you might be interested in. But I think most of the technologies start from the polyRNAs because it's kind of low-hanging fruit. If you use random primers, you can capture other RNAs. You can also, add a polyadenylation step, which is something that a later version of dbit-seq called patho-dbit does, and other technologies have done that, not necessarily just in the spatial context, but just in single cell context. So there are ways to capture other RNAs as well. Okay, and then in terms of the sequencing-based methods, well, I just mentioned, so even the probe-based method, it still covers the whole transcriptome. So the throughput is the same, but I guess, yeah, so in that particular case, it's using probes, but it's whole transcriptome. Yeah, and it doesn't add more time to have whole transcriptome. Okay, and then the strengths and not weaknesses, challenges of these capture and sequencing-based methods is, again, when it's sequenced, it can be untargeted and cover the whole transcriptome in a very high throughput manner. It's compatible with existing next generation sequencing workflows and a lot of more standardized workflows that maybe people are more widely familiar with. So it requires less specialized equipment. But the challenges can be that, well, you don't have imaging. So you're definitely going to have lower spatial resolution, even when the spot sizes are very high in resolution. So in many tissue types, you often find in the current state of technology that you can't necessarily resolve individual cells with super high confidence. As you have RNAs that are binding to your capture surface, you might have some potential RNA loss during that tissue removal. Alternatively, when you have cell-based, actually it's not listed here, but once you tag the tissue, you have your whole tissue, but during the dissociation, you might actually have some dropout of cells as well. When the RNA is actually diffusing onto the array, the diffusion might not just be, clear one-to-one, but actually like sideways diffusion as well. So that could also blur some spatial boundaries. But one thing I mentioned is that, the spot size isn't everything. So we might think that looks very, very high resolution. So for instance, stereo-seq, which has, I think 200-ish nanomolar resolution, they actually find that when they're trying to assign cells to or spots to cells, it'll be like 400 spots that are encompassing the cell, but because they have so many spots, then they actually have to bin it to be more inclusive of its neighbors. So I guess the spot size isn't everything in terms of resolution. And also, I guess this can be higher cost per sample, depending on the fabrication of the capture arrays. So if you're choosing a method, I think, a nice way to approach this is by, first defining your biological hypothesis and what is your actual question of interest. Is this more for, discovery? Are you looking for something new that maybe you weren't looking for? Or are you validating something? Or do you need to take a more targeted approach? And what are your gene coverage needs? So maybe you don't need to target the entire transcriptome, but maybe you do. What are your resolution requirements? Do you need to be looking at the subcellular level? Or is it more you're interested in like, general broad structures, in which case perhaps the tiny resolution differences actually really don't matter to you. And then I guess scale goes hand in hand with that. Another thing to consider is the sample type. So, a lot of these technologies actually require fresh frozen tissues, especially because RNAs degrade very quickly and need to be preserved correctly. And the moment you fix it, you're introducing a lot of these cross links that can be damaging. However, like FFP tissues, for instance, again, it's formalin fixed paraffin embedded. So there are these wax cassettes of all these archival human specimens across basically every pathology department, every hospital. It's so widely available. And is the preservation method like you don't have a choice in the type of fixation, or can you actually choose the fixation type because you are planning the experiment and collecting the tissues yourself? So I think the technology you choose might not have a FFP compatibility, so that is something to consider. And section thickness of what you want to capture, as well as the tissue quality of what's available. I think a couple other things to consider is, the actual technical expertise that is around you. And all of these technologies often require very heavy computational support or skills. I think some commercialized technologies try to streamline that for you, but that is definitely something that's not to be overlooked. And finally, there are very practical considerations in terms of available infrastructure. So is the equipment available? What are your budget restraints like? And do you want flexibility in what the technology does? So if it's commercially supported, it's going to be very expensive and probably a lot less flexible than if you're working with an open platform or something that a collaborator might have. So perhaps that is much cheaper and also provides greater flexibility. Okay, so in terms of future directions, so what's still hard is I just mentioned that FFP sample compatibility can remain challenging for actually like most of these applications. And if you can proactively actually fix the tissues yourself, that's very nice because you can control it. But often it's hyperfixed, so that's very difficult to get around. And in our lab as well as in many other labs, we're trying to figure out how to actually extract more RNA information, even if there are these archival specimens that we can't choose how they're fixed. The cost barrier still can remain high, and the computational demands, like I mentioned, can require a lot of work. And the other thing is, across all of these technologies, there's actually very little standardization. So, in terms of common benchmarks, I think it can be a little bit difficult to really standardize across all the technologies. In terms of what is coming up in the immediate to intermediate term, is technological advancements. We're gonna technologies are going to continue to become higher resolution. And one thing I kind of mentioned, but we didn't really elaborate on, is actually the capture area. So, a lot of these might be, millimeters big in terms of the surface area you're looking at. Up to 13 centimeters is, I think, what I have seen published. But I think scaling up the capture area consistently so that people can, not have to subsample their tissue to a very tiny area. The compatibility with FFP tissues, most of these protocols are incredibly time consuming. So how do we streamline that to faster workflows and increasing multi-omics capabilities so that we can really combine all the omics together to really scale this up and get more rich information? One thing that I'm personally interested in is pathology integration. So a lot of neurodegenerative diseases, or all neurodegenerative diseases have these protein pathologies that may not be inside, well, one, it's protein, and two, it may not be inside the nucleus, especially when you're using a dissociation-based technology. So, really integrating that and being able to capture, multidimensional information would be useful. And finally, I think there's a lot of computational advances that will be very exciting. So you asked about 3D reconstruction, so that's definitely in the works. Really studying on a higher throughput level, the cellular communication networks and their proximity. And finally, if you can use machine learning models to actually do feature extraction from images or just pattern recognition and spatial data, perhaps there will come a day when we have outputted enough really high quality data sets that we will be able to actually just image a cell and potentially be able to infer its transcriptomic state. So I think that is probably a slightly longer term, perhaps not so long because AI is crazy. But that is, what I think are exciting future directions. And finally, this is the last slide, is that I just wanted to say, there are all these technologies, but, from the history of, the foundations before the current era of spatial transcriptomics to now, spatial transcriptomics has come a really long way across all of these species and using all of these various technologies. You see all of these beautiful spatially resolved figures, and I just think that's really exciting, and I hope that you're excited about it too. This is just for further reading. If you're interested, this is not a comprehensive list at all, but it will be in the slide deck so that you can refer to it if you want to read more about any technologies. And thank you for your attention, and I will be happy to take any questions.
Thank you so much. I think because of time, we'll end here, but if you have questions, just please stick around. No seminar today, and we'll see you next week. Thanks, everybody.